Hi everyone, we're group 16 and we'll be talking to you about microbial communication and bioluminescence.
During our presentation, we will explore and investigate the background components and important concepts that are essential to understanding how microbial communication and bioluminescence work. We will also review mutations that occur as well as their significance. And finally, we will discuss the future perspectives and applications of bioluminescence.
The exam question we came up with is, what is the function of bioluminescence and how might it be beneficial or disadvantageous for a species?
I will start by providing some background knowledge on the Lux system of proteins and how they contribute to microbial communication and bioluminescence in marine environments. Bioluminescence, what is it? Bacterial bioluminescence is a good example of microbial group communication that involves individual microbial cells coordinating group behavior. Emission of light from a group of cells is highly effective and useful to organisms that have developed and evolved genetic material to accept these bacteria. These organisms use processes of bioluminescence for various behaviors in the environment, including defense, prey attraction, and intraspecific communication. How does it work? The emission of light by living organisms includes the development and maintenance of a genetic system requiring large amounts of chemical energy. Expression of phenotype is dependent on chemical cues from bacteria, and control of communication involves the accumulation of signal concentrations that cells process and respond to. Luminescent systems are complex, and specific genes are required to allow for the enzyme luciferase to catalyze the light-emitting reaction and use the high-energy substrates called luciferins for the final bioluminescent reaction. Light emission is described to be under strict regulation and involves various controls, including metabolism of energy, growth of the organism, and ability to respond to environmental signals that all contribute to the production of light. The Lux system. Let's keep in mind that bioluminescence is the process of light emission that results from an enzyme-catalyzed oxidation reaction in living organisms. The Lux system has the ability to produce a luminescent signal continuously or with a response to a trigger found in a large selection of organisms' hosts. All bioluminescent bacteria share the genes coding for the proteins responsible for light emission. The enzymes used are encoded in an operon with a single promoter with the core sequence of Lux C, D, A, B, or E. There are different theories as to how bioluminescence evolved. It was first assumed that the Lux operon and the effect of bioluminescence evolved independently at least 40 times. Another theory states the Lux operon arose once in the evolutionary past and that all genes have a common ancestor, with vertical gene transfer, duplications in genes, and gain and loss of recruitments, the systems of bioluminescence have overall evolved. Now we'll look into an example of Vibrio fischeri. Light production was first seen in the marine bacterium of Vibrio fischeri, which possessed genes responsible for light production. The Lux CDABE genes that are found on the second chromosome of these organisms form a part of an operon that allows for encoding the structural components that are essential for production of light. The main effect of light production is the enzyme luciferase, which is a heterodimer with alpha and beta subunits that encode for Lux A and Lux B. Luciferase is able to release light with the mixed function oxidation of a long-chain aldehyde and reduce flavin mononucleotide. Lux D uses fatty acyl groups from biosynthesis of fatty acids to use for luminescence. Lux C activates the acyl groups with AMP to reduce to the long-chain aldehyde by Lux E. Lux C and Lux E recycle this long-chain fatty acid from the luciferase reaction to reduce it back to an aldehyde. Lux G is shown to reduce FMN created by luciferase reaction. A very well-known organism that uses this microbe is the bobtail squid. It uses luminescence as a disguise as well as prey attraction and predation. These bioluminescent genes are linked to inheritance with quorum sensing that involves the chemical-based form of intercellular communication where bacteria are able to coordinate levels of population and community behaviors. Quorum sensing is associated with behaviors in bacteria including bioluminescence, production by Vibrio fischeri, and as these cultures grow, a signaling molecule, also known as an autoinducer, is collected within the media. Once a certain threshold is met, the concentration allows the cells to respond to the autoinducer by ultimately producing light. There are also regulatory elements that aid in autoinduction, including LuxI, that synthesizes the autoinducer molecule, and LuxR, that is a transcription factor and is autoinducer dependent. These enzymes, mentioned all, contribute to the core machinery of the Lux operon that allows for light emission and bacterial bioluminescence. Using structural properties of these enzymes of the Lux family and analyzing evolutionary conserved areas, a better understanding of these mechanisms can be observed and bacterial bioluminescence can be described.
Next, we will be discussing the important concepts.
Luciferin is the most important molecule for bioluminescence. Without it, there wouldn't be any light emitted. The process of bioluminescence starts when the protein binding substrate luciferin dissociates from its protein under external influences. These external influences are usually a disturbance in the water where the bacteria live. Then an enzyme called luciferase acts upon it in order to catalyze the reaction. Oxygen also plays a large role in this reaction, and the reaction impulse created from the reaction between oxygen and luciferin serves as a signal for its initiation. As a result, the cell membrane potential changes and the cell starts to glow. Some of the bacteria don't use luciferase, but instead use lucifins with oxygen and another agent, usually an ion of the element calcium, and this will emit the light. The reasons that luciferase is not required is because luciferase is just a catalyst, which allows the species to complete this chemical reaction faster. It's important to note that fluorescents are not the same as bioluminescence, since fluorescents are more like a glow-in-the-dark sticker, where bioluminescents are like a light bulb. The Lux operon is very important for bioluminescence, as it is responsible for coating all the parts necessary to emit light. Lux AB is required in order to produce luciferin, which I previously mentioned is crucial for bioluminescence. Lux CDE codes for the fatty acid reductase complex, pictured on the right, which is responsible for synthesizing fatty aldehydes for the luminescence reaction. Though there are a multitude of differences in the gene sequence and presence of other Lux operons that may be present. This causes room for variation from species to species. There are many different species of microbes that use bioluminescence in many different ways. This allows there to be a lot of variation based on a number of external and internal conditions. The most obvious factor that differs bioluminescent species is the color in which they emit. The three most common colors are green, orange, and red. The difference in color is caused by different variations in the protein luciferin. This doesn't allow the organism to change color though, as the color is generated in the cell's DNA. These three variations of luciferin are commonly found: D-luciferin, which causes the green light to be emitted; amino D-luciferin, which causes yellow or orange light to be emitted; and amino D-luciferin, which causes red light to be emitted. Though they are all different molecules, they all go through the same chemical reaction as I mentioned on the previous slide. And as you can see in the bottom left, those are the three colors with the three luciferins underneath them, and it causes that color to happen. If you look, it's not much a difference between each of the molecules, but there's a huge color change between all of them. The environment that they live in also has a large effect on how bright the glow is. In order to get as strong as the light as pictured on the right, there must be a large population of the bacteria dinoflagellate present. This is called a bloom and only occurs when there is a huge population within a small area. Sometimes this can be as many as 20 million individuals within a single liter. It was found when growing in a lab that bacteria strive when there is exactly 12 hours of light and 12 hours of dark within a 24-hour cycle. By doing this, they got the brightest bacteria that they could get. The salinity, depth, and current of this water also has a large effect, as a large number of bioluminescent bacteria are found in shallow, salty bays protected from the ocean current.
Next, I'll be discussing mutations. The first mutation I came across was one that enhances bacterial bioluminescence. The luminescence emitted from an organism can fluctuate from high to low brightness. In this study, they generated a new lux operon called iLux that increased the brightness by 7 times. They were able to identify multiple mutations in Lux A, Lux B, Lux C, and FRP. In the table below, you can see the various types of mutations they found that were capable of increasing the brightness. They found no mutations in Lux D or Lux E that enhanced luminescence. This study was significant because it created a tool to see single cells in E. coli with increased clarity. This opens up possibilities for observing various processes such as death and division of single cells. It also opens up possibilities for testing the effects of new antibiotics because scientists will be able to examine the bacterial at a single cell level, thus providing more in-depth information regarding antibiotic resistance. The other mutation I found was a study on non-bioluminescent Vibrio fischeri. It has been discovered that there are non-bioluminescent strains of Vibros that possess Lux genes, which are the primary genes that are responsible for bioluminescence. A study by O'Grady and Wimpy found 3 dark genotypes that cause these mutations. The genotypes of the mutants consist of an insert sequence, point mutations, and deletions. The 3 mutations that caused non-bioluminescence were an IS10 insertion sequence in Lux A with a mutated Lux E stop codon and a truncated Lux H gene. The second mutant genotype consisted of a deletion in Lux C and lastly a frame shift in Lux C. In the picture below, you can see the difference between a non-bioluminescent bobtail squid and one that's bioluminescent. They were able to find these mutations by screening over 600 Vibrio species.